Hello, GraphConnect. Good morning.
Good morning.
So we have lunch after this, right? So this is probably a good time to talk a little bit about food and where food comes from and how technologies like graph databases are very effective tools at doing that better. So like Ryan said, my name is Tim Williamson. I'm a data scientist at Monsanto. Monsanto is a global agricultural products company. We develop productivity products, mostly seed, that are sold in about 160 countries worldwide. I've been with Monsanto for 6 years. I'm greatly privileged to work on a small team of ragtag engineers and data scientists that have been tasked with coming up with really novel ways to get better and more effective inferences out of our genomics pipeline. And that's what I'm here to talk to you about today, how we've achieved that in some bite-sized chunks. Like I said, I've been with Monsanto 6 years. It seems like an eternity now, but in a former life, I was a scientist at the Department of Biomedical Engineering at Washington University in St. Louis. And one of the reasons why I made that transition from the comfortable cocoon of academia to the industrial world is I became very aware that our planet faces some very big challenges as it grows to what's projected to be over 9.5 billion people by 2050. A lot of people to feed. And the expanding urban footprint that comes along with that number of people is increasingly coming at the cost of productive farmland. So people want to live in cities. They don't want to live in rural communities anymore. They move to cities. Cities have to expand, and that land has to come from somewhere. So much so that it's projected by the time that we hit that population level, there'll be less than 1/3 of an acre of productive farmland left to feed each of those people. In addition, that growing population moving to cities is also continually entering a more global definition of middle class. And one of the many things that comes with being middle class on the global scale is a shift to using sources of animal protein more and more in your diet. So people want to eat more meat, eggs, and dairy. And that's important because it is more resource intensive to produce those products, which further stresses the agricultural production system. To top it all off, I think everyone in this room is aware of the strange and dramatic effects that global climate change is having on our daily life. And in particular, as far as making severe weather both more frequent and harder to predict. And I put that in context, and I think an example that how many people here are actually native to California? All right, so is it too sensitive to talk about water availability issues? I don't know, sensitive not? So to put this in context, in December 2014, a group of scientists at NASA published a very extensive computational model projecting that in order to truly end and recover from the California drought, it would take about 11 trillion gallons of water spread evenly over the state to do so, which is a multitude more than the largest reservoir in the world. Now, that volume of water is the exact same volume of water that over the course of 5 days in the first week of October was dropped on North and South Carolina, if anyone remembers that news. So all the water that you needed was somewhere in the world, but it wasn't where we needed it. And part of all that destruction was that flood occurred during the middle of peak harvest season in those states. So now, this is not, so let's try to raise up a little bit. This is not all doom and gloom. Humanity actually has quite a few tools that we can use to address these problems. And one of those tools in particular, which is improving the genetics of staple crops, we have several millennia of experience doing. So approximately 10,000 years ago, the first farmers began the process of transforming teosinte on the left, which is the wild grass ancestor to modern corn, into the super optimized solar-powered calorie factory that we know today. And they were able to do this because they made two key observations. So one, when they looked across a field of teosinte, they observed that some of those plants displayed more highly desirable traits, such as producing more seed, than did others. And second, if they selected those plants and only replanted them next year and let them repollinate each other, then over time, the frequency of plants which displayed those desirable traits would go up. So fast forward several centuries, that act of choosing to plant and allow to reproduce one plant or population over another because of something that you want to see is called artificial selection or selective breeding. And when that accumulates over centuries, you can develop new crop species or end up with the same type of crop species that's been more optimized to do things like feed people. So in the modern world, so we've jumped forward from history to the modern world. In about the mid-1800s, the USDA, the United States Department of Agriculture, began regularly compiling nationwide yield statistics for important staple crops like corn. And that's what you're seeing here, is the yield performance of corn nationwide in the US every year. Now, for those who don't regularly deal with agricultural units of measure, a bushel, so bushels per acre is how we measure grain production in crops like corn. A bushel of corn is about 56 pounds of corn seed. And an acre of land is about 90% the area of an American football field. So, and one thing that's really obvious looking at this is from the 1860s to about the 1930s, the improvements in corn had largely stagnated. So we weren't moving the needle very, very quickly. And that would be particularly depressing, except that humanity didn't sit idly by and wait another several millennia to introduce a step change in productivity of that crop. So analogous to the way that advances in modern transistor fabrication technology have allowed CPU processing speed to keep pace with Moore's law, we've had several big revolutions in genetics and our understanding of how genetics affect traits that have allowed us to introduce large rate changes across that curve that have allowed gains to keep pace with population. So examples of that, going from starting in the 30s and going forward, were we figured out how to hybridize certain crops, make new varieties. We discovered molecular biology. Molecular biology happened during this time period, and we discovered exactly how to isolate particular pieces of DNA that are directly correlated with performance. And we very recently, in the past 10 or 15 years, became very good at making very targeted modifications to those crops to get exactly what we want. So what I'm here to tell you today is that the next rate change in that curve is actually going to be one borne out by a completely genomic data science-driven pipeline. It's not going to be our ability to collect more data. It's going to be our ability to make sense of that data in a meaningful way quicker. And right now, we are in the process of reconfiguring all our global crop breeding pipelines, which span 8 different commodity crops and about 18 different vegetable crop families, in order to take advantage of some of the advances I'm going to talk to you about today. So we're pretty excited about this. So where does genetic gain actually come from? So how do we actually do this? So regardless of whether you're at a university lab or a corporate lab, the way at which you develop better crops largely revolves around this concept of a breeding cycle. So a breeding cycle begins when a discovery breeder picks 2 parents, a male and a female, each of which is chosen because they both have some set of traits that we want. So one's maybe resistant to a disease, the other grows really well in a certain soil type, and we want to come up with one progeny, one plant that has all those sets of favorable traits and nothing we don't want. So the way that happens is we cross those 2 parents and we generate what might be hundreds of thousands to millions of progeny, and we pass them through a breeding cycle, which is a highly focused and optimized device of selective pressure. At every stage of the cycle, we are continually looking and screening for only the plants which have everything we want and nothing we don't. So select the best, discard the rest. And the winners at each stage are allowed, we plant them, we let them pollinate each other, and then their progeny are advanced to the next round of the cycle. And at the very end of a cycle, our goal is to come up with one new plant that displays everything we wanted about both those parents, nothing we didn't. And that plant will go on to be used in future crosses, or will frequently end up in a product that's sold to a farmer somewhere in the world. Now, at every stage of that cycle, we generally rely on 2 classes of data set in order to make the decision on what we're going to keep and what we're going to discard. So one data set is generated in a lab, and it's called genotype data. So today was a big day for 23andMe in the news. I don't know if you're a genetics nerd like me, you've been following this all day. Does anyone here ever had their 23andMe marker panel run? Oh, come on, this is San Francisco. All right. So exact same principle here. So when we're screening for genotype data, we're looking for a set of genetic facts about that plant that are predictive of what it's going to perform like. And this is cheap and easy to collect for us. Later on, we also reach a point where to understand complex traits, we have to plant field trials and grow them and actually measure how they perform. And unfortunately, this is fairly expensive and time-consuming because you have to plant, you have to have people to go collect the data, monitor their trials, and so on and so forth. And as an example of scale in our pipeline, we may every year at that step make thousands of crosses, take dozens of progeny that we plant across 5 to 10 locations apiece at the cost of several $1 million in order to gather this data. This represents a bottleneck in our pipeline. Well, what we want to do is get much better out of every cycle, getting a slightly better plant. And really, there are 3 ways that we can go about doing it, broad classes. One, we can get more predictive about which are the 2 best parents to cross. So you might have hundreds of thousands of parents. How can you use data science to figure out which are the best ones to cross together? 2, at every round of the selection pipeline, how can you make the best possible decision? So not an okay decision, not a terrible decision, hopefully, but the best possible decision. And 3rd, you could try to expand this pipeline. So simply test more stuff. However, to throw a wrinkle in this, we pretty much are maxed out on the land that we have available for field trial usage. So if we want to expand the width of the pipeline, we somehow magically have to do this without planting any more trials. So hold that thought for a few slides and think about just that progeny, that one plant coming out of the bottom of that funnel. So now we're going to start to look a little bit more graphy, a little bit less genetics, a little bit more graphy here. So C represents the one plant that came out of that cycle. And it has an unbroken tree of ancestors. So these singly linked operations represent those self-progeny generations at every step of the cycle, linking back to the 2 parents that were crossed to start its production. The length of that chain can grow approximately 4 hops a year as we are converging to whatever the best possible parent is, their best possible outcome is. And that, especially if that endpoint, if that end parent that comes out of that line ends up being really successful, it might go into hundreds or thousands of its own crosses. Add on top of that, we've been a plant breeding company for about 20 years, and we have about 60 years of data going back before that. So each one of those parents, A and B, has its own rich history of ancestors that were crossed to yield it. Over time, this builds up. So again, let's take a look at parent C and imagine it in the center of that data set. So that is a real data set. In the center of that is the oldest known source of a really critical corn line in our R&D pipeline. And over time, that line has been so important and been such a good parent that it's been used in hundreds of thousands. There are at least 300,000 plants represented on that graph, and I don't think that's even everything, spiraling out of it. So that one parent in the center, its genetic impact is felt all across our pipeline. And if we want to make a step change in those 3 areas that I told you about genetic gain, essentially they all run through the fact that to make the inferences that we want better, we have to be able to mine data sets of this size for very complex and meaningful genetic features, and we have to do it in what approximates real time and at large scale. That's our problem. Well, I said that we have been a plant breeding company for about 20 years, and we have about 6 years of data, and this is all long before the concept of a graph database even existed. But we somehow had to make this work, and we did this by shoving this ancestor data into a relational data structure. I'm not going to beat the dead horse on this because I think everyone in the room, if I asked you to make a set of nested join tables that would approximate a graph as best you could, you would all know how to do it. You should not do it, but you would know how to do it. In our situation, our pre-existing relational stores encompassed about 11 or so of these really nasty join tables, representing about 900 million rows of data. And as you can probably imagine, every single read from this system is a fairly unpleasant exercise in connect by prior. So massive recursive join. Now, if you are particularly overconfident Oracle DBA, you might say that this is a problem that can be solved. It's not that bad. Fortunately, we're scientists, and we can measure this. So the equivalent of hello world in questions of genetic ancestry is, if I give you a plant or an animal, if there's a mammalian pipeline, if I give you something, I want you to return to me the entire tree of all its known ancestors, just everything. I don't want you to prune it, just give me back the tree. So we were doing this at Monsanto. We had optimized our tables the best that we could. wrote the most optimized recursive join nightmarish query that we could. It was production optimized, used all sorts of production caching on a very massive Exadata environment that we run. Obviously, our definition of real time can vary upon who you're asking, but there's nothing about that performance curve that says real time to me. So as you're advancing across the x-axis, depth is hop up the tree. So one level of ancestral depth. And the response time is measured on execution time of returning the data set back. And this, if you dig in, this precisely correlates to when the data set starts to explode in number of ancestors. Now, the reason why, if you're wondering, we only went up to 15, though there's hundreds of levels of depth in this tree, is that by level 15, we went to lunch, and we came back. It was a long lunch, 90 minutes at our favorite Mexican restaurant, and it was still spinning, so we figured we learned everything we needed to know at that point. Well, what do? We still have to solve this problem. We're still fighting every day in production, wrapping our heads around this. Well, fortunately for us, after much wailing and gnashing of teeth, we realized that genetic ancestry forms a naturally occurring graph, if we allow it. And what you're seeing here is a somewhat small, trivialized snapshot of how we actually handled this problem. But I think if you step back and think about what your family tree looks like, you would know exactly how this would look in a graph. And indeed, that's what we ended up doing. So every bag of seed has a node. We track with a new node every time it's planted somewhere on Earth. Anytime something is harvested out of that plot, we track it. And it allows us to build a really nice abstraction layer from parent to progeny as deep as we want to go. And as an estimate of scale, when we first built the final test run of this graph and stood it up, it's about 700 million nodes, growing every day, about 1.2 billion relationships, and almost 2 billion properties. So I don't think this is a particularly gigantic graph, but it's also not a trivial small graph. So now, going back to where we started, so again, we take our same production SQL, production Exadata instance, and we recreate the exact same algorithm using Neo4j's traversal framework, using the exact conditions and data sets that we're going to run this in production, because that's the only reliable benchmark in the world, and compare the two. Pretty cool, huh? This probably doesn't surprise anyone in the room, but now this is a completely scale-free problem for us. We can now process these arbitrarily depth trees at what approximates a completely scale-free performance. And what's neat about this is not the fact that we can do it quickly, it's that now that we can do it quickly, we're allowed to do two things that were really holding us back. One, the number of use cases where you want to see all the parents of the tree isn't particularly relevant from a data mining perspective. You want to find a feature. But if you're always fighting with performance, we can't build abstractions that start to get at those features, which we now can. And two, I don't want to analyze the ancestry of one plant. I want to analyze the ancestry of 1 million plants, and I want to do it within a few minutes. That's what we have to be able to do. So from this point forward, we're going to leave the what was world, and we're going to go into what is now, and talk about how we built those abstractions that allowed us to start wrapping our heads around these data science problems. So, and just to kind of leave on this, again, we didn't see a need to take this out past 15, but that's a 90-fold performance difference. So it's a game changer. Now, our philosophy on delivering data science products in our group is that it's not sufficient to build a super cool data store, throw it in front of a bunch of data scientists, and say, learn how to query it. Does anyone work in a company where you do that? No one wants to operate that way. So what we set out to do is how can we encapsulate that data set in terms of a really common biological grammar, accessible by REST, so accessible by whatever programming language you want to do. That way we can execute really complicated algorithms, but we don't have to teach anyone how to do it. So a fairly trivialized example is think about a really basic tree. So population 1 has one singly linked ancestor. It breaks off into a cross. Each cross has its own singly ancestor. Really basic tree. In our grammar, we encapsulated returning that entire tree with a simple REST resource that says, hey, I have population 1, give me all its ancestors. And we return back to the requester a fairly cogent JSON representation of a tree. Every node's represented, every relationship's represented. So what that means is that if we have someone that's really pushing the limits on some very one-off algorithm that we don't want to implement ourselves, they get an entire subgraph from Neo in real time. So they get effectively the power of our giant graph without copying the entire thing over to their machine, which obviously we don't want to do. But that would have been boring to stop there. Again, I kept mentioning important genetic features. So there are a bunch of milestones in genetics that are important for understanding and maximizing the information that you gain out of a lineage. And we wanted to write grammar that allowed scientists to access that. So a very basic one is, if I am plant 1, I want you to show me, I don't care about my tree, but I do want to know when is the most recent binary cross in my ancestry. When did I have 2 parents? So that's really important because genetically that represents what's called a recombination event. The time in my lineage when 2 parents with different chromosomes were crossed together to yield a heterogeneous population. It's huge for genetics work. Well, in our system, all a user has to specify is give me the binary cross. And they'll get back the exact male and the exact female, and they will go about their way. Now, these 2 paradigms can be combined together really cool. If you think about, all right, what if I really am interested in that feature, but I still want to know the path that I took to get there? Well, that works too. And the way we conceive that in our grammar is, all right, I want the ancestors of population 1, but I want you to prune that tree and give it back to me when you recognize the first binary cross. So in that case, we return the same tree structure, but now if you notice, it's been pruned at the level of that binary cross. So we set about making every conceivable, we kind of broke out the old graduate school genetics textbooks and conceived of every meaningful pattern we could think of and built a grammar around it. And in September 2014, which is auspicious because we were flying out to GraphConnect shortly after that last year and hoping that everything would be rock solid, we turned the lights on the system. And at Monsanto, this is an internal system, we called it Ancestry as a Service because everything has to be as a service now or it's not cool. And what that really is that rich, RESTful grammar over an embedded Neo4j cluster that contains the entire genetic history of our pipeline. And that wrestler is what's directly accessible by an ecosystem of both individual data scientists and application developers. As it stands today, there are about 30 elements of RESTful grammar in that system that allow you to cut our pipeline from the oldest known discovery cross to the most recent product that's being sold on a field today. There are about 120 applications and data scientists that are using the system today. And because a lot of those are applications, they serve their own user base. So again, maybe not impressive if we were a web company, but these are all internal scientists. And so far, they've made well over 600 million REST queries on the system. And remember, each REST query here is its own complicated graph traversal algorithm that's running behind the scenes. To put context how this really changed our world, most of the applications built over this platform didn't exist before we did this, but one in particular, one application that touched every seed in our pipeline, observed a 10x performance increase when it cut over. And one particular data scientist was able to replace 1 month of analysis time with 3 hours of crunching by porting his code to work over this system. Now, real-time reads require real-time data. It would be insufficient for us to say the system is real-time if we have stale data the minute we build the graph. And we dealt with this problem by recognizing the fact that we can't direct every application writing into our monolith to start writing into the graph, not without, again, much wailing and gnashing of teeth. We solved this by utilizing a technology that Oracle sells called Golden Gate, which lets you generate a change capture stream from the redo logs coming off an Exadata instance. And we wrote a custom adapter that pipes that change stream and transformed it into something that Apache Kafka can recognize. And that change stream is what continually feeds our graph, such that we ingest about 10 million writes a day, and under normal conditions, within a second or two of that write hitting Oracle, it's reflected in our ancestry layer. And this paradigm is actually one that we're not just using here. We're using this to slowly slice apart our entire monolith and get it out into more appropriate data structures off of prem, off of our data centers into AWS. We think that this is really very useful for companies of our size and in our situation, so much so that this morning we open sourced this. So if anyone wants to pull down this adapter and see how they can start breaking apart their monoliths in a really modern way, please do so. Ask us questions about it. We're really proud of the work. So we're going to use the rest of the time to step into the future here. So we figured out Ancestry, but the next step for us has always been the fact that this diagram, which we call the Triforce, represents the holy grail of modern genomic science. What traits do you observe about a plant, its phenotypes, indexed against what do you know genetically about that plant that's predictive of that performance, partitioned by what environment, what stress factors did you measure that performance in, all linked together by Ancestry. Blows open the doors on quite a few really interesting genetic conundrums that exist in modern science. So one of those problems is actually accessible by considering how we actually layer genotype data onto our ancestry graph, which is really easy to do in a schema-light system. We attack that by saying, okay, for every plant that we have a set of genotype data on, we're going to build a simple node linked to that population, and that node acts as a simple pointer to an external system that stores all of our genomic data, in our case, an HBase environment. And what that lets us do is then start building algorithms that parse that ancestry graph based on the presence of certain genotypic features. So we go back to a trivialized example. What that looks like is, so I have 3 entities in this graph. They each have a genomic data set linked to them. The property of marker count directly correlates to the size of the data set. So 60,000 markers would be 60,000 facts that I know. 300 would be fewer facts. The numbers are so different because it's very expensive to gather large marker data sets. So we only do it on things that we're going to use as parents. We can't afford to do it on every million of progeny that we generate. But we can generate really small marker data sets on those progeny. And it turns out, and this would be a talk all of its own eventually, is that there's some well worked out math that if you have the location of all your marker data sets, your low and your high density ones, and the exact genetic path between those data sets, so between progeny to parents, you can actually upsample the data content of your low resolution data set to be pretty close to what it would have been to measure the expensive data set. So if we wanted to build a system around doing that, in our RESTful grammar, that takes the form of, I am this population. Now I want you to prune the tree when you find the most recent set of genotype ancestors and give me back those data sets. In this case, those IDs represent the keys that link out to our HBase data store that has those data sets, which means now that is a tree that's annotated with all the presence of the genomic data that we know about those populations. Well, how that works in practice is really cool, and I hope that we can talk more about this in future months, is that, so now that we can spit out these genotyped annotated ancestry trees from Ancestry as a Service, we had a group of colleagues, one of which is here today, that built a Spark and GraphX-based genotype estimation engine over that system. So it consumes a tree of ancestors linked with genotype data sets, and it upsamples, it estimates the high-resolution genotype of the target progeny. And when it does so, it pulls in the data sets it needs from HBase using those linked keys. It writes the estimated genotypes back to HBase, and at the same time, it communicates to our graph via Kafka. That way we can take advantage of those new data sets in future calculations in this giant feedback loop. And what this does very much, like today, going on right now, is gives us the power to estimate at a very high level the genotype of every single seed that goes through one of our breeding pipelines. Now, why does that matter other than the fact that it's cool? Well, you remember how I said earlier that we're really limited in our ability to expand horizontally, or we can't horizontally scale our pipeline very well at all unless we get really, really clever about how we use data. In this case, because now we have high-resolution genotype data on all those seeds, we can replace that first step of field trials by estimating those genotypes and mining that data genetically for the most effective progeny. So make the selection. And what that results in is better selections down the entire breeding cycle. So that's our bullet point 2. But at the same time, it's really quick and easy to screen these in a lab. So no field trials are planted, no months are consumed. It's less than $1 million a year to gather that data, which effectively frees up land to be used elsewhere. So we expand the horizontal scaling of our pipeline by virtue of leveraging that data. And that's been pretty phenomenal. We're pretty stoked about this and doing this across every crop that we have. So I'd kind of like to go towards the end here by really kind of sharing some more random thoughts that I have about where we're going next. So I am a biologist by training. I've kind of ended up I've taken a weird path to get to where I am now, computationally. But it fascinates me how geneticists have known about the power of graph analysis for a long time. So that left-handed example, I was going through an old textbook, and that page comes from a poultry breeding textbook from 1921. So remember, 1921 is pre-molecular biology. We had no idea how to describe the heritability of traits very well on any very meaningful descriptive level. But the scientist has drawn out this structure, this graph structure, that represents for any progeny way down the tree how all of its ancestors contributed to genetically, just by virtue of crossing. That's really neat. It turns out that what he's describing is a set of algorithms called Coancestry analysis, which actually form the basis of a very powerful class of genomic selection techniques. That paper that I show is from 2010, is, again, a great paper that describes a series of graph-based algorithms using that theory to make really informative selections on what parents you cross and what progeny you pick out. Again, no one in this ecosystem has any idea what a graph database is, yet they're getting at it. So that's what we're going after. So we are very close, in the case of Coancestry as an example, of producing these reduced representation coancestry matrices that describe for any given input, any given plant, what contribution genetically each one of its crossing ancestors makes to it. So it's not foreign to graph theoreticians to have a matrix form of a graph. It's a very efficient way to compute. So we are going to be spinning up some of these genomic selection algorithms, again, powered by these graph-based data sets, to try to further turn the needle on, okay, let's not just make better selections, let's not just widen the pipeline, let's put the right stuff into the pipeline in the first place. And this has been really exciting for us to do because we can now. So I'd like to end here with a couple thoughts. One, there are many people who contributed a lot to get me on this stage right now, but I would be remiss if I didn't thank two of them in particular, which are Jason Clark and Marshall Marietta, that are both here right now. They are the other two members of what we call colloquially around our company GraphRoots, which is kind of the ragtag team of people that went off and just started doing all of this, started trying to reinvent the way that we manage these data sets. I'm active on Twitter. You are happy to reach out to me at any time, and we can talk about genetics or graph databases or graph algorithms, or that's pretty much about it. I like dog rescue too. We can talk about rescuing dogs. If you'd like to, in a few days, I'll have a version of this talk in long article form published on engineering.monsanto.com if you want to take a look at it there, or take a look at anything else that's going on with some of my teammates. And at this time, I'd love to take any of your questions, or if you don't want to talk out loud, we can talk at the side. Oh. All right. So how many people are in this situation where they have a giant monolith of data and they need to split it out? Oh, come on. There's no shame here. All right. Rob, yeah, I know you. So the reality is that in a large functioning company, it would be really enticing just to say, turn off all the applications and start riding to the new sexy data source. But most of us get paid to keep the lights on, right? So coming up with this flow lets you have both. You can build the sexy new data store, redirect everyone to read from it, but at the same time not compromise your rights coming in on the other side. And that lets you slowly to carve off giant chunks of these monoliths without turning the lights off. Anyone else? No, he's running now. Now you got to use the mic. It'd just be insulting otherwise.
All right. When you say that, I got the mic. You and a couple guys broke off to form this GraphRoots team. How did you go about kind of organizationally approaching that prospect?
We should definitely have a couple beers and talk about that. But I think the three of us all worked in different parts of our R&D pipeline, essentially battling this ancestry issue. So me from a data science perspective, Marshall and Jason from doing some really core engineering work. And we had an epiphany at the same time that this was awful, and we couldn't actually do anything of significant scientific value. So it actually started by we took one day at work. We get a free day every couple weeks to do whatever we want. We stood up a really promising prototype in Neo4j. And then for the next couple months, we largely, on nights and weekends, we kind of locked ourselves in a room and built out this new store. All the entire data store, most of the foundational algorithms, and then we essentially pitched it as almost like a separately funded company. And we asked for the money to put it into production. And it worked. I mean, that was... Would it have worked had we not actually gone off and done all that? I don't know. I don't think this is the kind of work that could have been funded very easily in a traditional project proposal, but I think now we're at a place in our company where people believe this stuff is possible. So the next round of stuff that we want to do and we will be doing and are doing right now, we have very little resistance to. So just do it, I guess.
Other than Ancestry data, is Monsanto using like graph databases for any other projects that you know of?
Yeah, so this work, our success here, we've largely seeded a couple other use cases in a couple different areas that we're trying to get off the ground. So one is, like many companies, we struggle with identity management and identity asset provisioning. So that's definitely one case that we're working on. In another, so what I showed you there was a hierarchy of genetic data. So these plants have these ancestors. But in an actual R&D pipeline, there's another layer of data over that is, what decisions did every scientist make? And how did that cascade down? So the decision layer. That is another really valuable data set that we have some colleagues that are trying to, actually they are in production with essentially a version of the graph that tracks every decision made in our pipeline. So I am this scientist, and I chose to select these plants and move them forward. So we can go back and actually monitor whether that was a good decision and better make predictions on what are the right decisions. Those are two really good examples. Well, thank you, guys. That was fun. Thank you for being such a good audience.